Good evening. Today we are going to discuss about intracellular accumulation, which is the last topic of cell injury, cell death, and cell adaptations chapter. So, as the name indicates, intracellular accumulation refers to accumulation of different chemical substances in cytoplasm, nucleus, or in any other cell organelles which are present inside the cell. Such intracellular accumulations can be observed in any error in metabolic processes or during any pathological conditions. We might experience intracellular accumulations. Next, let's see what are the chemicals, possible chemicals that could be accumulated inside the cell to cause intracellular accumulation. Those substances include in lipids, proteins, hyaline cartilage, glycogen, pigments, and calcium. Next, let us see about lipids. In lipids, uh, there are many three chemical components, that is triglyceride, cholesterol, cholesterol esters, and phospholipids. Okay, so these three chemicals are found when there is any accumulation of fat inside the cell. So we'll see the pathological cases one by one. First, let's see what is steatosis. Steatosis refers to fatty change. Okay, that means abnormal accumulation of triglycerides in normal parenchymal cells. Okay, since liver is the predominant or principal site for fat metabolism, steatosis is usually common in liver. Though steatosis occurs in liver, but not only in liver, but also in other parts such as kidney, muscles, and heart muscles. Okay, in all these three, we will also you can encounter steatosis. And then the main causes of steatosis include any side effects of toxins or due to diabetes mellitus or due to obesity or due to protein malnutrition. We might encounter such steatosis condition in patients. So the most common cause of significant fatty change, that is the most common cause of steatosis, is nothing but alcohol abuse patients. And if you see alcohol abuse people, steatosis of liver is more common. But apart from that, there are also other steatosis of liver. There is non-alcoholic steatosis or non-alcoholic fatty liver disease, which is associated with diabetes and obesity. Next, let's see what happens if cholesterol esters accumulate. So we know that cholesterol is an important chemical component for synthesis of lipids or for synthesis of cell membranes. Okay, but cells ensure that there is no accumulation of cholesterol and cholesterol esters. But in certain pathological condition, cholesterol accumulates, especially in the vacuoles. So let us see one by one detail. These are the four clinical conditions which are caused due to accumulation of cholesterol. Okay, so first let us see what is atherosclerosis. So in atherosclerosis, atherosclerotic plaques, we will see about smooth muscle cells and macrophages were situated in the inner layer of aorta, and it has large arteries filled with lipid vacuoles. Okay, so most of which are made up of cholesterol and cholesterol esters. Next, cholesterol and cholesterol esters. These cells have foamy appearance. They produce the yellow cholesterol laden atheromas, so which is the characteristic of this disorder. So in atherosclerosis, we will observe that smooth muscle cells and macrophages within the intimal layer of aorta and large arteries are filled with lipid vacuoles. That is a very important point. And then uh, actually due to the rupture of fat laden cells, these fat laden cells due to rupture of fat laden cells, cholesterol and cholesterol cells are leaked into the extracellular spaces and form crystals. So they form the long needles that produce cleft in tissues, whereas other small crystals of lipids are phagocytosed by macrophages and activate the inflammasome, contributing to inflammation. So this is the pathology behind atherosclerosis. So hope you all understand atherosclerosis. Next, I'm moving on to xanthomas. What is xanthomas? Is nothing but it is also like a cluster of foamy cells which are present in subepithelial connective tissue of the skin and in tendons, which produce tumor known as xanthoma. So xanthomas is nothing but intracellular accumulation of cholesterol within the macrophages. That is what xanthomas. Okay, these have become foamy cells, and they're usually located in the uh, they're usually located where? Usually located in subepithelial connective tissue of the skin and tendons. Okay, that is very important location, and the cell, which cell is undergoing this macrophages. Next, if we talk about cholesterolosis, cholesterol is nothing but accumulation of cholesterol-laden macrophages, as you seen before in xanthomas, the same thing, but this time accumulation occurs in the laminar propria of gallbladder. That is what the difference between the location of accumulation is different in xanthomas. In xanthomas, it is nothing but subepithelial connective tissue of skin and in tendons, whereas in cholesterolosis, such accumulation is taking place in lamina propria of the gallbladder. Next, let's see what is Niemann Pick disease. This is a very important question which may be asked in MCQs or in short notes. So, Niemann Pick disease type 0 is type C. This is nothing but it is also a fat accumulation disorder only. But here the lysosomal storage disease is nothing but Niemann Pick disease. What happens is the mutations or caused which will affect the enzyme involved in cholesterol controlling, cholesterol regulating amount is lost due to mutations which will affect an enzyme that is responsible for cholesterol regulation. Okay, and what happens is the finally excess cholesterol will get accumulated in multiple organs, causing Niemann-Pick disease. That is what about Niemann-Pick disease. So we need to remember that this is a lysosomal storage disease, a very important word term. Next, let's see what happens if proteins accumulate in our body. So due to intracellular accumulation of proteins, they appear in histological slide as eosinophilic droplets, vacuoles, or even as an aggregate in the cytoplasm. So let us see what are the pathological conditions occur for protein accumulation. The first one is reabsorption droplets in proximal renal tubules. See, in case of renal diseases, due to associated protein loss in urine. So, in nephrotic syndrome, in first year we have learned that presence of protein in urine is an indicative of nephrotic syndrome. Or if a protein is present in urine, it is also referred to as, the condition is also referred to as proteinuria. In kidney, so very small amounts of protein are normally filtered through glomerulus, but they are reabsorbed by proximal convoluted tubule through pinocytosis. Okay, so like in certain disorders with heavy protein leakage across the glomerular filtration barrier, what happens is the proximal convoluted tubule will show increased reabsorption of such proteins, excess proteins into vesicles, and in our slide the protein appears as pink hyaline droplets. Okay, so if proteinuria is there, definitely we will see this. We will see this spots. I think we will see this uh, spots like vesicles like structure which are hyaline like, and then these droplets will disappear when the proteinuria ceases. Then uh, so the proteins usually accumulated in our cells or usually normally secreted proteins, but that may be produced in excessive amounts. Okay, so that is what about first point. Reabsorption droplets in proximal renal tubules. Okay, next let's see about what you mean by Russell bodies. Russell body is also important. What happens, as we have seen before, that in endoplasmic reticulum, the proteins will get accumulated. Excess proteins will get accumulated. So what happens is certain plasma cells are involved in active synthesis of immunoglobulins. Okay. Okay, so that is so the endoplasmic reticulum becomes very stressful condition, and then they will produce, as a result they produce large homogeneous eosinophilic inclusions called Russell bodies. Next, let us see what happens in alpha 1 antitrypsin deficiency. In alpha 1 antitrypsin deficiency, the mutations which occur in the protein significantly slow down the folding process, resulting in building up of partially folded integrals. Proteins will not get, okay, will not show proper folding, that they will be misfolded proteins only, and these proteins will aggregate in the endoplasmic reticulum of hepatocyte and that is in liver cells and aren't even secreted. Especially if there is a deficiency of alpha-1 antitrypsin where it is needed in the lung, which causes emphysema. That is what the resultant deficiencies in circulating enzymes are needed. That is, enzymes are not at all transported properly. In that case, one example is there, alpha-1 antitrypsin is nothing but a protein, but it is not transported properly in our body. So, the lungs will not get enough amount of this circulating alpha-1 antitrypsin enzyme and causes emphysema. So, in many of these protein-folding diseases, the pathology results not only from loss of protein function, but also endoplasmic reticulum stress. That is what accumulation of misfolded proteins will cause endoplasmic reticulum stress. Okay, that is very important, and it will automatically, finally, these misfolded proteins will automatically lead to cell death by, they show signaling to cell death by apoptosis. So next, I'll be discussing about accumulation of cytoskeletal proteins. So here also what happens is similar to only microtubules and then actin filaments, myosin filaments, and intermediate filaments are accumulated. So intermediate filaments usually provide a flexible intracellular scaffold that organizes the cytoplasm and resist the forces applied to the cell and are divided into 5 classes: keratin filaments, desmin, neurofilaments, minimetin, and glial filaments. Okay. So you have to remember the names of the cytoskeletal proteins, and then alcoholic hyaline is an eosinophilic cytoplasmic inclusion in liver cells, which is an indicative of liver disease. Okay, that is very important. And neurofibrillary tangle, which is found in brain in Alzheimer's disease, also contains neurofilaments and other proteins. So you have to remember the examples. For example, alcoholic hyaline, okay, which indicates the alcoholic liver disease, and then neurofibrillary tangle, which is found in brain in Alzheimer's disease, containing neurofilaments and other proteins. That is very important. And then aggregation of abnormal proteins. See, abnormal proteins, as we seen earlier, it causes endoplasmic reticulum stress and interfere with normal functions. They can be intercellular, extracellular deposition of both. Okay, so certain forms of amyloidosis fall in this category of diseases. We will see later. And then these disorders are sometimes called as proteinopathies or protein aggregation disease. That is what about aggregation of abnormal proteins. In the next video, we will be discussing about hyaline cartilage, glycogen, and pigments in detail.